Alicia Campbell joining. Sorry, Alicia Campbell, PhD candidate of Molecular Cell Biology and Neuroscience from Rowan University. Alicia, you can explain that more than I can. So we're just very grateful that Alicia has raised her hand as a volunteer to join our community and kind of help us with the knowledge about genetics and MED13L. And we just thought this was like a really great place to start to see where our community is and then see where the questions and the conversation goes. And after today's Zoom, if we feel like we need to do another one or a different discussion, we'll meet with Alicia and try to come up with something. So with that, I want to get right into Alicia's presentation. And thank you again, Alicia. We really appreciate this as a community.
Yeah, of course. So I guess to introduce myself, I am going into my 4th year of my PhD program. So I have about technically 2 more years left. And I'm actually the person who works on the samples that were taken up in New York last year-ish. So I am working on the cell lines. I'm working on collecting data from those, interpreting it, putting it all together. And then collaborating with a bunch of other people in the community that do a bunch of different type of experiments. So I thought it would be good to kind of start with really the basics, just because so we can grab everybody in this net and keep moving. So I'm assuming we have a variety of backgrounds as well. I'll leave it open to questions at any time. So if you want to put something in the chat, or if you just unmute yourself, I won't be offended if you stop me mid-sentence, because a lot of these things, raise a lot of questions. So, okay, so first off, we can take a look inside of the cell. So this is obviously what makes up the human body. There are many different types of cells, and they have all different purposes. So I've highlighted here lung, liver, skin. This is something that's pretty general knowledge, right? So all of these, depending on where they're located, have different functions. And inside the cell itself, we have different compartments that are responsible for different functions. So as 2 examples, I have the nucleus. That's where our DNA is. And we also have mitochondria. So this is known as the powerhouse of the cell, which everybody loves to remember from high school biology. This is where we get all of our energy from. So actually a closer look inside of the nucleus itself is where we find our DNA, like I said before, and this is found in the form of chromosomes. And just remember, this is inside of the human cell. The next picture I'll bring up here are actually 2 karyotypes. So this is what you would get done normally during an amniocentesis if you opt to have an amnio. This is all 46 of the human chromosomes for both a female on the left, male on the right. The distinction is your, let me put my laser here. The distinction here is your X and Y chromosomes, which we all know. XY is going to be male, XX is going to be female. So there's 23 pairs otherwise. And all of our DNA is basically everything that we need is inside of these guys right here. So what exactly are chromosomes other than these kind of string bean looking things? They contain our genetic program. So you can see if you unwind the chromosome a bunch of different times, you can see all these different letters here, which is what we'll call our genetic program. DNA has to be packaged like this because it's super, super, super long. And what it is actually super coiled. So if you imagine twisting a rubber band on itself a bunch of times, you get one coil, and then eventually you'll start to get a second coil on top, and a third and a fourth, until you can no longer twist that rubber band. So basically it's packaging everything together in a really tight, compact environment. In terms of the DNA itself, what we think of typically when we look at this is the DNA double helix. A little bit closer, I have a picture here. We see all of the letters that everybody associates with DNA, the A, T, G, and C. And our bodies actually have tiny cellular machines that are needed to read this sequence of random letters and decode it. So we go from one message here to another message down here. This is known as mRNA, and mRNA serves as our intermediate. You might hear mRNA recently in the news for all of our COVID vaccines. So it's actually just the strand of letters here that are making up the code for these vaccines. But our bodies go from DNA to mRNA to eventually protein. And protein is exactly what you think it is. When you think of a protein supplement or your protein shakes you take before you do a workout, it can consist of glycine, serine, alanine, or a couple that people generally think of. So for more of a simplified version of this picture here, we go from DNA. Our cells read this. They use this as instructions to make protein, and proteins are the building block of the human body. So we need all of these proteins to be fully functional, to be properly made, properly read in order for the body to function. So proteins themselves have a lot of different functions in the body. And in our context here, these are just some of the examples of what proteins can do. MED13L itself is a protein. So it helps the cells function. It helps the cells carry out daily tasks. For example, MED13L or MED13 is needed to help regulate DNA and RNA expression, so making more of those initial building blocks. It's also needed to provide support to the body, so this is when we think of muscle and muscle strength. But it's a lot more complex than that. These are just some examples for now that I could think of in terms of what a protein actually is. So take a second to review what we have so far. So we have cells making up the body. Too many to count. It's a lot of exponents on the cells that are in the body. They each have specialized functions. They contain a nucleus with the instructions in the form of DNA. These instructions are known as genes. And then all of these instructions are unique, so the cells have their individual specialized functions. And lastly, genes are the code for proteins. So how do we get different proteins? How do we get different genes? This is something that a lot of people, when we dive deeper into genetics, a lot of people have questions on the actual, well, how do we read this? What does it mean? What does it mean for me? And how am I different from other people? So different versions of the same gene are called alleles. And the easiest thing to think of here is there is one gene for eye color. This gene has many different variations or versions of itself. So you have a blue version, you have a green version, a brown version. Another diagram that you see in genetics textbooks are genotype and phenotype. So genotype, these are going to be your alleles. These up here in this first box are your alleles. So they're all the same gene, and you can see the repeating pattern of C used in all of these. But there's different outcomes depending on the combination of C and its exponent that you have. So that's the relationship between your genotype, so what's actually encoded in your DNA sequence, versus your phenotype or your physical appearance. So to work our way through this, if we have two capital C's, we're going to have a black fur rabbit. But if we have lowercase c's with this exponent of CH, you're going to have chinchilla. So this is just the same version of explaining how we have brown eyes versus blue eyes versus green eyes. Up here, you would have maybe B, the letter B for brown, blue, etc. And this would lead to a different outcome in the eye color. So how do we get these? And this is kind of where we can go into inheritance. So everybody inherits things from their parents. there is a caveat to that, but I'll get to that later on in terms of inheritance. So we have mother, we have the father, and your children are a mix of the mother and father. So that's kind of how you can say that a kid is a combination of your parents. And you say, oh, maybe it looks like more like the father, more like the mother, because kids really are the mixed bag of both parents. You get half of your set from your mom, half of your set from your dad. And depending on the gene, and I'll go through terminology on the next slide, depending on the gene, there's different outcomes for our phenotype. So that's our genotype-phenotype relation again there. Yeah. Do we have a question or no? No, okay. All right. So kids are basically, again, recap, kids are a combination of the parents. Here's another example. So this is, you would have the father here in blue, mother in pink. And this little black box is going to serve as our gene. And both the mother and father contributed. So your mother contributed this version from her chromosomes. Father contributed this version. So if your father has brown eyes, mother has brown eyes, your child is going to be a combination of the chromosomes that they inherited. So they're going to have brown eyes. This slide I titled genetic jargon because there's a lot of different terms that are used in genetics that can be confusing and sometimes need a little bit more explanation. And the colors actually match. They're in little pairs, you'll see. But so in terms of autosomal or somatic, so this relates to the entire body and it can affect both sexes equally. So either male or female can inherit it, and it's going to come from either mom, dad, or it can become a spontaneous mutation, which is from neither mom or dad, but it is just a completely random mutation that the child receives. We also have germline. So this is from either the X or Y chromosome. This is how we have the difference of the two sexes. So it's not going to affect both sexes equally, such that if you have, the easiest example to think of here is with color blindness. Color blindness is typically associated with males because they only have the X and the Y. The mutation for color blindness is found on the X chromosome. Since they are only going to get one copy of X, they don't have that other X to compensate like females do, so they have an X and a Y, hence the color blindness is mainly a male phenotype. We also have recessive and dominant. So recessive means it takes two mutated versions of the gene to show signs and symptoms. So this is both mom and dad are contributing. So it's either you basically need both, one from mom and one from dad in order to show signs and symptoms. However, dominant, either mom or dad can contribute to the phenotype of the offspring. And lastly, to bring back in our genotype phenotype, because it's really important to kind of drill in this whole relationship between your genes and the outcomes of the physical appearance. So genotype is going to be if you have a missing letter in your genome, if you have another letter in place. Phenotype is what we actually see as a result of that genotype, so the physical effects. So if your body has the code for blue eyes, you're going to show blue eyes. Same thing with curly hair. If you have the gene for curly hair, you're going to have a phenotype, so your physical characteristic is going to be curly hair. So probably something that a lot of people are interested here is, what is MED13L syndrome considered in terms of genetics? So it is autosomal or somatic, so that means it affects the entire body. It is dominant, so that means that it can come from either the father or the mother, or as I mentioned before, it could be spontaneous, so neither the father nor the mother, but just something that happened along the way. So not necessarily the parents contributing. The genotype for this would be a mutation in chromosome 12. And this would be either your gene is missing, your letters or your DNA are mixed up, the protein is not made properly. And I'll go more into the actual DNA mutations in the next couple of slides, because I'm sure people have heard, you know, my child has a frameshift mutation or a deletion mutation, but they're not necessarily sure what that really means in the context of all the different types of mutations. So phenotype, again, this is what we're more familiar with. I just have a couple examples here, but there's a bunch of them. I always like to point out that this is a syndrome, so no two people who have MED13L syndrome are alike. Everybody is unique. So in terms of bringing this back to a summary slide that incorporates kind of the same messages as before. So we have the human cell, the nucleus, our chromosomes, which we talked about, and now bringing it into the case of MED13L syndrome. So if you do not have a mutation in your MED13L gene, you're going to produce a version of the MED13L protein or MED13 that is functional in the form as it's able to carry out its daily tasks in the cell. If you have a mutation, this here is signified by a little break in the DNA. The next slide I'll have the specific mutations, as I promised. If you have a mutation, you're going to have a version of MED13L that maybe isn't able to carry out its functions in the cell, or it's hindered in some way, or it's just different from this other version up here. So it might not necessarily mean that the protein is completely non-functional, but it just means that the actual sequence, when you compare to between these two, the sequence itself is different. So what exactly is a mutation? The big thing that I like to drive home here is that no matter what the DNA mutation is, it's going to lead to a version of a protein. So normally proteins are able to be formed, be made from the DNA, and carry out their functions in the cell. When you have a mutation, you're going to have a version of the protein that comes with a variety of outcomes. So it's not necessarily what we associate when the protein is made in full, or the sequence is exactly as we would expect. So the first type of mutation I have here, this is kind of small, but we have a deletion. So you can see this little piece of DNA was here, but it left. It's no longer there, such that when we're going to read this new sequence of DNA that comes out, we have a disrupted sequence. So this is kind of like if you're trying to read a book and somebody deletes or rips out a chapter of your book. You're going to read it as you normally would, but since this chapter is deleted, the end of the story is going to be different. So you're going to miss what's in that chapter that got deleted or ripped out because you're not having the full story. We also have point mutations. This is where we have one letter that is accidentally switched for another letter. So here in the case of C, this was highlighted just that maybe this was an A or a T or a G, and now it's accidentally a C. So this is just one small point mutation in the DNA. We also have a couple of different frame shift mutations, and they all lead to different outcomes. So if we have a frame shift mutation that is resulting from a deletion, we are missing, you can see here, you are missing a U that was in the sequence, and now the entire sequence is going to read differently. So if you're missing maybe all the vowels in your novel that you're reading, your story is going to read a little bit differently. We also have a mutation where a couple of letters are missing, so not just one, but we have maybe 3 or 4 or 10 that are missing. And this leads again to the entire sequence shifting, so hence a frame shift. And lastly, we have what is called an insertion, where an extra base pair or an extra letter is inserted into the sequence. So this just happens when we're copying the DNA, making more DNA, making more cells. Something goes on where we have an extra letter that's accidentally added into the sequence. So what causes these DNA mutations? This is normally one of the biggest questions that I get, how did this happen? What is going on in the cell that made this happen in the first place? So sometimes the answer is nothing. And as I mentioned before, this is called a spontaneous mutation, or these mutations are also known as de novo in the genetics terms. So quite literally, sometimes the cell just makes up its own mind and creates a sequence of DNA that is different than what it was originally. So there is no known cause. So like I said, this occurs during the process of making more DNA. So the machinery itself is just accidentally misreading the sequence. We also have external factors. So this is typically in the case of breaking down DNA. This can include environmental toxins, radiation. The best example I could think of here was damaging UV rays from the sun. These actual mutations from the UV rays lead to skin cancer. And it, you probably didn't expect it, but it's really just a plug for wearing your sunscreen at the beach. But this is something that is less likely the case in MED13L syndrome. We're more so looking at spontaneous or random mutations, or this last one here, inherited mutations. This is from any relative. This could be mom, dad. It skipped a generation. It came from grandmother, grandfather. And in terms of MED13L syndrome, they are really across the board. So we have cases where it is de novo mutations. We also have cases where it's inherited. But it is important to just point out that there are other ways you can get DNA mutations. However, this is not the case for MED13L syndrome specifically. And lastly, I'll kind of go into the molecular biology of MED13L syndrome. So what's actually happening in a cell in this case? So MED13 and MED13L are part of this complex of 4 proteins. It's known as the CKM, or the CDK kinase module. And basically, it's a bundle of proteins that come together inside of the nucleus of the cell and work together for a certain outcome. So these other proteins, cyclin C, MED12, CDK8 or 19, and you also notice that MED13 and MED13L are interchangeable. So some of the cells in the body have MED13, some cells have MED13L. It just depends on the system of the body. It depends on the pattern of inheritance. And this unit as a whole is necessary to help with the production of other genes. So when I talk about reading the DNA sequence and making more proteins, making more genes, this is what MED13, MED13L, and the CKM module is needed for. I pointed out a set of genes here, the mitochondrial maintenance genes. So these are just helping the mitochondria function. So like I said earlier, the mitochondria is the powerhouse of the cell. So it's where all the cell's energy comes from. So if we don't have a functioning CKM, we don't have production of these mitochondrial genes. This is a really complicated diagram that actually shows all of the other MEDs. So MED stands for mediator complex. There's roughly 20 to 26, depending on the species, the organism. Here we have what I just had on the previous slide. So MED13, 12, cyclin C, CDK8. But you can see that it's actually part of a larger complex with all these other MEDs. This is needed to communicate signals. So from outside of the cell can interpret the signals and provide a set outcome, so it can help relay signals and interpret them. It helps transmit signals to regulate gene expression. So if we need to upregulate stress response genes so the cell can respond to stress appropriately, that is where the mediator comes into play. So again, with information about how genes get turned on or turned off, we need the mediator complex to help relay signals from outside the cell, interpret these, and help the cell elicit the proper fate. I did mention earlier the mitochondrial genes. So these are needed for the functioning of the mitochondria again. This is the diagram that I actually use when I present any of my MED13L work. So this is kind of the working model that we have in our group specifically. So we have hypothesized that when we have a non-functional version of the CKM, so basically we have a mutated form of MED13L, we don't have our mitochondrial maintenance genes, so we cannot upkeep the mitochondria. This is going to decrease a bunch of things related to the mitochondrial health. And this is just a bunch of diagrams, but what this really means is in terms of neurological function. So we have neurons that heavily rely on healthy mitochondria. We also have muscle function. So hypotonia is one of the symptoms of MED13L syndrome, depending on the case and the severity. With muscle, we need obviously a lot of mitochondria. And in terms of development, so if we don't have proper regulation of development-related genes, so this would no longer be mitochondrial genes, we would have development genes. If we don't have those being made, this could be a way that developmental delays or intellectual disabilities come into play. And lastly, I'll show you some of the mutations that we've seen in MED13L syndrome that we've seen so far. So if you can recount the mutation slide that I showed with all of those different types of mutations, I will pop up a bunch of these here. And you can see that just for the ones, these are a couple of the ones that I study, you can see that they're all really all across the board. There's no two mutations that are alike. There's no that are located in a specific region of the MED13L gene. So my goal as a scientist now is to take the type of mutation, so the location on the MED13L gene, and the type, so whether it's a deletion, a nonsense mutation, missense, frame shift, whatever it is, and if I can correlate this with the outcome for the patient, so how can this be predictive of what the children experience in terms of their phenotype, so how severe the phenotype may be or lack thereof. So if I can take all of the genetic information and make predictions about what the phenotype or what the outcome would be. If you have any questions, I do have my email up here, and I'll just leave this up here. Unless somebody needs me to go back to another slide, I can certainly do that. For general science-based questions, I'm not a genetic counselor, nor can I advise on, future pregnancies and stuff like that. That I will 100% defer to somebody who's licensed in genetic counseling. But if you have any questions about science or what's going on in a cell, what does DNA mean, what do any of these things mean for my child, I can certainly answer those questions. So I did provide my email here. And I'm sure Kelly, Kim, everybody at the foundation also has my email too, if anybody has any science-related questions. But I can also take some time now to answer any of the questions that you have about this presentation or any kind of lingering genetic-related questions that you might have had based off of a consultation in the past. So anything like that I can hopefully answer.
I have a question when you're ready, Alicia.
Yeah, no, I'm good. Go ahead.
Okay. So I have an 18-year-old, almost 19-year-old. She's going to be 19 actually in about 2 months.
Mm-hmm.
My question, when you talked about developmental, you had mentioned intellectual.
Mm-hmm.
What I find really interesting too is that they're because, and I'm speaking from a parent of someone who's older, I know a lot of these children are younger. she's not really meeting the developmental steps in puberty in a
Mhm.
normal sequence. So like she got her period, I think around 12, which maybe is a little bit later. Actually, maybe it was 13 maybe. She got
Okay.
it. Not like anything major to be worried about, but there was no breast development. And one of the things that I wanted to mention, I actually got her ultrasound by a breast specialist, a
Mhm.
radiologist who's, just to make sure everything was okay. And there's actually like an absence of breast tissue.
Okay.
So unless she's a candidate for, constructive cosmetic surgery, actually, because she actually doesn't really have, she has a very, very small, it's like maybe the size of a quarter of breast tissue.
Sure.
And I think that's really interesting, being that she's literally hitting 19 years old, but has had her period since about 13.
Right.
I thought I would just mention that because we were talking about developmental and
Yeah.
does actual like puberty changes. And they said this is so unusual because usually you see breast development in girls, but then you'll have delayed menses. But in this
Right.
case, it's kind of the opposite. I also see some hirudism, which is interesting, and I would like to eventually look into that.
Yeah, no, I don't think it's completely out there, especially because a lot of the mediator related genes, so MED13L, it specifically controls development related genes. So whether this is in utero with the embryo itself, it does control development related genes there, but it also controls developmental factors that are needed for later in life. So whether that's bone formation as the children grow older, or in your case, this would be related to puberty or lack thereof of certain parts of it. It honestly does not surprise me at all that some aspects of development are affected.
There's another thing I wanted to ask Alicia, and I apologize to the team that I'm taking all this time up, but being that my daughter's one of the oldest, can you discuss your knowledge of mitochondrial we know that the brain uses a lot of mitochondria, but I believe kidneys do also. My daughter's cardiologist is well known, and he understands mitochondrial disease quite well, actually, and has personal experience with it. And after speaking with him about it, asked for a full panel, including BUN and creatinine levels, which are now elevated. And I find, not like horribly, but she's just off the scale in the elevated section at 19. And when you compare it to like 2 years ago, it's increasing every year, or it's at least increasing. I would say that was bi-yearly. But we're going to be keeping a closer eye on that. So I just wanted to mention that.
Yeah. So that's actually my specialty before I fell into the hands of MED13L syndrome was mitochondrial related disease phenotypes, diseases, how the mitochondria are regulated. So I come from a mitochondria specific lab. And actually a lot of the work that we've done relates to cell death, how we can utilize cell death for actually treating cancer. So we were originally a cancer lab until I kind of dipped my toes into this with Randy, but I'm not 100% sure about the mitochondria in kidneys in particular. I am very familiar with mitochondria in neurons, and I do know that that is probably why we're seeing some sort of neurological symptoms, or at least I'd like to hope to say that's why we're seeing neurological symptoms. But in terms of the kidneys, I don't know if anybody has actually put out kidney panels, so in terms of blood work, and whether that correlates. So I don't know if anybody has actually done, I'm curious to see if any other parents have done kidney panels, kidney blood panels as well, and if that's common across.
Yeah. I
Okay.
will post, I will go online when I have a chance and post mine to Simmons Searchlight.
Yeah.
I haven't done it yet, actually. I've just got busy, but I doubt it, because being that my daughter's 19, I actually had to push for those blood tests to be done. They were not, doctors were not readily offering them to me. I actually had to say, I had beat around the bush and finally got the support of a cardiologist.
Right.
Who said, yeah, maybe that would be a good idea.
Yeah.
Just hopping on to that, we actually did, my son is 3, and we actually did like a renal panel. I'm looking at it right now, and his BUN was normal, but his creatinine was low. So just for some insight on what you were talking about.
Yeah, that's definitely something that, you know, as parents come across different symptoms, honestly, anything is helpful in terms of being able to correlate if this is common among everybody or if this is just a subset. Any piece of data really helps with collecting knowledge on MED13L syndrome.
I have a question related to like the mitochondria and like the neurons. I know like you had said that could be a reason for neurological symptoms. And with like kids developing at different rates, is there a way to like test that specifically for each child to see like what that function is? I know, you know, like say our son like was delayed in walking, like does that relate to the amount of delay that you'll see progressing through life?
Right. So I know one of the groups right now is working on neurological based samples. So the samples that I have are just the skin punctures, the biopsies. But there is actually a way that we can transform some of these into neural based cells, and then do kind of exploratory on those to see what is going on in terms of development, if this is related to the actual neurons, the projections of the neurons, where all the different links are in the brain, if this is different in some patient samples versus other patient samples. That's technically beyond my scope, unfortunately, just because I am only dealing with the direct primary patient samples. So I don't have the technology to make them into the neurons, but it is something that I believe is on the up and coming for other scientists, in terms of looking at specific neurons or creating MED13L mutations in neurons and looking to see how these neurons function in the lab setting, and whether this is, you know, predictive of another way that you can go with this is you can look in rodent models. So you can look at how either mice or rats, they can do kind of, there's a bunch of different tests that you can do. They can do grip strength, so how long a mouse is able to hold onto a bar before it falls off. They can do different, what they do, they stain the bottom of their paws as they walk along a piece of white paper to look at their gait, so how they walk. So if we're able to create mice, and I know one of our collaborators has or is in the works to create mice, to see how these develop. So starting from embryo all the way through the full life of the animal, how this affects different stages, male versus female, that it is all in the works.
Hi, I just wanted to say I'm Marcy and I'm from Australia, and my daughter Grace is 22. And just to the other lady that was saying about the breast tissue, she has one very, very small breast and one non-breast. So she's like, you know, half and half on the body. And that body seems to be, that side's a little bit weaker as well, that has the no breast. But I just wanted to find out, she has started probably the last maybe 6 months, and I don't know whether anybody else has noticed this or whether this is related, and you might be able to help me with the mitochondrial. She's got an appointment with a neurologist coming up, shaking a little bit constantly like that, but it's not visible. It's sort of more visible if you're near her or sitting near her, like she's sitting on the couch next to you, you can feel her constantly moving. Would that be related? And she's about developmental wise 3 or 4.
Okay.
So she didn't walk till she was like nearly 11. She talked maybe 12 or 13, started saying a few words. We did Makaton and sign language before that. So her vocabulary is like a 3 or 4-year-old, maybe even a 2-year-old sometimes, but comprehension is fantastic. So
Yeah, I
what do
think
you
that's
think about
definitely,
that?
yeah. In terms of the shaking, I know one of the common symptoms that people see is hypotonia, so the loss of muscle,
Yes.
mass, muscle weakness, so that may go hand in hand with that. That might also be, in terms of shaking, that might be something neurological, which I'm sure your neurologist has plenty of answers
When we
related
get
to that.
there.
Yeah.
But because it's so rare, even him looking at her, yeah, I mean, well, this recording, maybe I could give that to them or something just to understand, because in Australia, nobody's heard of it, nobody knows what it is, and any doctors you go to, they're all like, oh, what's that? And they start looking it up. So, yeah, it's interesting. And in saying that, exactly what you just said, as she's gotten older, she's gotten weaker. So once upon a time, she could ride a push bike and run and stuff like that. And now she can walk maybe sort of 10 minutes and has to go in her wheelchair. Very, very weak. Very, very weak.
Yeah.
Yeah.
I think that's something that also a couple of the parents that I've talked to, they have also experienced that same issue where it's almost like bursts of energy. And then they start to kind of trail off and need to rest for a while. So that is another common thing that's definitely coming out of the woodwork now.
Cool. Great. Thank you.
Alicia, Vanessa Diaz here. So with that, thank you, Marcia, for sharing that, because that actually reminded me I wanted to ask on the Zoom, because I know a lot of people probably have this question where, as you were saying, like children will gain skills, and as they age, we're hearing from certain members of our group who have older children that their children are starting to have these new issues, and what they once had, they're losing it, or they're starting to experience these random things where it just seems like they're suddenly a lot more complicated than they used to be. Is it something where, like, you know, obviously with our children, they have this MED13L genetic variant. Is it where at some point, because it's so different that the production of whatever mitochondria it could produce just starts to really kind of give out, is because it just seems like as our children are getting older, I'm just reading that, you know, the older kids are just really starting to just have different challenges. So what's the thought process behind that?
So that's actually one of my more recent projects, is looking at in the cellular models, what is happening as these cells, in my case, get older. So as they live in my little plates that I have, so what happens if I test the mitochondrial function at a later point? So if I let these go out a couple weeks, what happens to these right as they're towards the end of their lifespan? So cells only actually will live until about 30 doublings. So as the cells split, they can only split 30 times, roughly. So one of the things I'm looking at is if mitochondrial function is declining. And I've seen a couple of patient samples where it is over time. I've seen other patient samples where it's the complete opposite. So it goes up, and others where there's no change in the mitochondrial function. So that's kind of up in the air right now. But that'll be something that'll hopefully eventually come out when I am able to put all of the data together. What is going on in terms of the so-called cellular age. And I'm sure Chad will look at the mice and how they do throughout their entire life progression. And if there's some sort of, even if it's like muscle weakness that progresses with age, or if it reverses with age as they learn new tactile skills or motor skills. So it might be a case-by-case basis, but it definitely, I'm trying to make a connection right now with mitochondria and what I can say about mitochondria and how that relates to the cells overall.
Thank you.
Yeah.
I guess I just want to introduce myself to everybody. I think I've met a lot of people already on here. My name is Vanessa Diaz. I am on the board of the foundation, which honestly, in the grand scheme of things, means nothing. I'm just another mom of a child with MED13L that's trying to push for research. So, and so the rest of the board is made up of that as well. So, don't feel like you can't connect with us. You can't, contact us or ask questions. We will share as we can. Alicia was brought into our life through Dr. Strich at Rowan University, and as she so wonderfully, thank you, Alicia, for dedicating your Sunday evening to helping our community understand what's going on. I know for a lot of us, that don't have the medical background, and even myself, I'm a registered nurse, but even like once you get that diagnosis, like all your knowledge of anything just goes out the window because you're too busy grieving, right? So it's a huge process, but having this extra breakdown of a live person explaining to us what all of this really means, I think is just it's priceless. So thank you so much for that. I want to say on here, we do have a lot of stuff going on within the foundation. Again, we have to, I guess, consider how we release things or whatever, but it's just so much is going on in such an exciting way. There's a lot of momentum. So know that myself, Kelly, Kim, Katie, and Nick, we are all like pushing, and we're pushing for fabulous people like Alicia to, help us figure out what's going on with our kids, what we can do to help our kids, what makes our kids different, even within our own community. As Alicia mentioned, there are things going on. We just funded a study at Vanderbilt where we're going to be looking into that. As Alicia mentioned, we have researchers that are going to try to differentiate the difference between all those different variants that Alicia explained, like the one where it's just one letter that switched, or if it's like 3 letters that switched, or if it's like a big deletion. We're having a researcher figure out, does that make each one of those unique to itself, to those group? Are those deletion kids in a group? Are the 1 letter kids in a separate group? We need to figure that out, because when you look at the types of treatment we're looking at, if we're looking to fix the MED13L variant, we need to know what one treatment is going to do for one and what it's going to do for the other, whether it works or it doesn't. And then what's exciting about what Alicia is doing for our community is she's helping us understand the downstream effects of what this genetic variant is doing to our kids, right? And so what is super exciting about that is there's so much research going on already with other groups where they have things in clinical trials and stuff potentially on the market, I don't know, Alicia, for mitochondrial dysfunction. So, we're still trying to figure out what's going on with MED13L. We're trying to figure out, repurposed drugs to help boost the MED13L. We need to make sure that those compounds will are going to be safe for everybody, which is why we need to make sure that the deletions are the same as the 1-letter switches or the 3-letter switches. We need to make sure that everyone is the same, because we don't want to boost somebody where their information is coming out and it's the wrong information, and that wrong information is what's causing their complications. So we want to make sure it's safe to give that group that could be having the dominant negative, right? Where it's like, the information is coming out and it's really causing complications. We want to make sure that they're going to be safe, that those kids are going to be safe to try this medication. So all of this stuff, I know it probably seems like we're so disconnected as a foundation. Ashley, you were phenomenal and connecting with us and saying what's going on. So we appreciate it. But know that it seems like in order to figure out what the main dish is, we have to figure out what the ingredients are that go into that dish, and then where did those ingredients come from? Do you know what I mean? So it's all this stuff that's happening behind the scenes, and we're just parents, just like you. I have 3 kids. I have a separate job, so that the MED13L Foundation is obviously like a passion for myself, because my youngest has MED13L, and I think a lot of you on this call have met her. So we're fighting, we're exhausted, just like the rest of you guys. So, just if you have any questions, feel free to send an email to us. We'll connect you with the right person if we don't know the answers. And if sometimes we don't have the right people that know the answers. So just keep in mind, we're just parents too, so just thrown into this new world that just like the rest of you. So we appreciate your patience. We appreciate everyone jumping on this call tonight, and definitely Alicia for taking her Sunday evening to educate our community, which is huge. So does anyone have any questions for me?
I have one question, actually for you and Alicia, but I'll ask you the question first. When the drugs that can be repurposed are established, or the whole MED13 world is ready for it, is there talk, or could there be, everyone goes to doctors that don't know what's going on with MED13, and I feel like when we go to doctors, we're kind of like bringing them up to speed. If there's ever a drug that comes out that would be safe for everyone, is it possible to get a doctor that would kind of oversee all of the MED13 patients that are taking it or not, so that they're kind of like more experienced or have more expertise with it?
Yes. So that's a complicated answer. And that was my first thing too. And, especially with like telehealth during COVID, everyone was able to like see doctors that were, 3 states over or whatever. But those days of kind of like the Wild West medicine for COVID during the COVID days, it's over. So physicians can't actually treat outside of their licensed state. So every physician is licensed. Some of them, like where we are, I'm in New York, a lot of them are licensed in the tri-state area, because we're just kind of like, our 3 states are kind of like a community, because everyone goes from New Jersey to come work in the city, and then Connecticut, people go work in the city as well, in New York. So there's this collaborative thing that goes on with our states, but I think that's unique to our 3. So what's really, really exciting, though, is because what would be nice, and I want to say that that's actually what was promised to us, or was told that was going to happen when we first joined Rare Base. But I think that they had wanted to do it too. And I think during the time with COVID, it was something that could happen, because doctors were allowed to treat patients from different states, just given the scarcity of everything. But now that all of those, the Wild West is over, what we found out is that Dr. Wendy Chung, who I think all of you guys know, if you don't know, look her up. She's on our scientific advisory board. She is a phenomenal woman who is, like, to me, like the queen of genetics. She is going to be heading over to Boston Children's. and which it's sad for me because she's usually just 30-minute drive from me, but she's going to Boston. But what Boston Children's is doing is something super cool, because we went to the Global Genes Conference recently where we met up with all sorts of rare disease groups, and everyone kind of had the same issue where it's like our community is so small and our community is so spread out. We can't get the funding or pick even the location as to where we can have a center of excellence, right? Kelly and I have talked about that a lot because she and I are both CHOP lovers, right? We love going to Children's Hospital of Philadelphia. I love that institution. But it doesn't, we approach them as a foundation to see, like, hey, can you guys do like a dedicated day, even if it's just once a year, where you can have a geneticist, a neurologist, a cardiologist, a developmental neurologist, a dietitian. Everybody goes there on one day, and then we kind of go see the physicians in like a round robin fashion, because they do that for different conditions. Like my older son has eosinophilic esophagitis, which is short-term EOE. That's what it's called. They have an EOE clinic where it's once a month. They have the specialists of the EOE, the allergist, the gastroenterologist, and a dietitian. I go to one appointment and I sit in the patient room with him. And those 3 practitioners just come in and speak with us. And then they collaborate outside. And then the allergist comes in and tells us the plan. So that's what I envisioned as like our dream. I know that's what Kelly wants for our community. And I'm like, why can't we do that? So I emailed CHOP and said, hey, can we do this? And they said, yes, but you have to donate, like, I think it was like something ridiculous, like $100,000 or $200,000 a year. We can't do that. We can't do that. We want to fund studies, like, in the meantime, like we need to like figure out what's going on with our kids first. Because as you guys know, the specialists are very important. But at the same time, they're not the day-to-day, right? How many times do you actually call your neurologist unless your child is having like active seizures? You see them like once a year, right? How many times do you call your cardiologist unless you're going in for surgery for the heart defects after that? You're just monitoring once a year. So for us to donate that amount of money to one institution for like one day, like even once every 3 months, we just can't do it. We have too much stuff we want to fund that's actually going to find therapies for our kids, right? Find drugs for our kids. So a bunch of people were saying the same thing at this Global Genes Conference. And so Boston Children's raised their hand and like, we're going to do it. So Wendy Chung, who's amazing, she's going to head that up at Boston Children's. So it is her passion to create this specialist center where it's going to be, she's going to create this like force of nature team of the neurologists that are specialized in rare neurodevelopmental disorders, right? So these people, they may not just be MED13L specialists. They're also going to be like SYNGAP1 specialists. Like their kids are very similar to ours. So these are going to be the physicians, like the cardiologists or like the orthopedists. Like it's going to be a team of professionals there that are used to seeing rare. And so their goal is, when they go there, hopefully they get all this information from all of our communities and they can really help us from top down how to figure all this stuff out for our kids. So what I think is going to happen, don't hold me to it, but what I think is going to happen is once it's all established, I think we're going to suggest to families to start, like, make a trip once a year. And I'm not saying that right now, guys, don't book anything. It's just once Wendy Chung gives us the green light of like, this is what we can do for your families, then maybe we'll do our family meetups there. And I think that's maybe a way we can piggyback things, is like the families come together, we all do our days of like medical evals, and then we hang out as families. Because I mean, I know Kelly and Kim know, and Ashley knows, and everybody on this call who's like been to like the Baltimore thing. It's so insanely comforting to be around these other families. It's so, like, you look at these kids and you're like, that's my daughter. They're so similar. Or you look at these boys and you're like, that's my son. They're so similar. And so to me, that's my dream, you know, as a parent for the foundation, is to have it where everyone can kind of piggyback our family meetup and see the doctors all at once. And then we can all like create this place where we have these doctors that are like experts on MED13L, right? Because that's what we need. We need the experts, like you're saying. So the other thing, Ashley, is I like, especially because we're recording, I can't like say some of the things, but you know, we're working, it's literally, we're achieving things that are going to make it a lot easier to identify our community. Do you know what I'm saying? We're achieving things where it's going to be internationally identifiable. So when somebody gets, you know, like, oh, I don't know what to do with this kid, they can plug it in and, you know, they'll be able to say, oh, these are, it's okay, she can come. They know what to do. There's going to be direction given to them, you know, so they're not going to feel so lost. This is my daughter. Oh, okay, where are you at? Oh, I'm blurred, honey. Hold on. You want to say hi? This is Al. All right, she just
Vanessa, you won't believe how much she looks like my daughter.
really?
They look like twins. I'm not joking.
Isn't it
put
crazy?
a photo in the Facebook.
Let me see if she can come back. She's trying to boot my husband out of here, but sorry. This is like recording our family craziness. You want to say hi? So this is Al. Can you say hi?
Hi, Elle. Hello.
Hi, your friends. Yeah, so that's it. And, you know, there's some exciting things coming up for us to share with the community. We're just not quite there yet, but just know we are working really hard, and stuff is happening. So, you know, the biggest thing that we need as the foundation is community participation and community fundraising. Because all of these things, you know, like Alicia, you see how her lights are on in the background, she needs to be able to pay for that. So she is, I'm sure, as much as she loves figuring all this stuff out for MED13L, she needs to get paid for the work that she does. So that's really where all of the fundraising comes from. You know, we're not, you know, none of us are taking a salary. You know, it's at this point, we are, we're all volunteers and we're all parents just like you. So.
Okay.
What, Vanessa, sorry, what about if you live in other countries? You wouldn't be able to get probably to Boston to do that sort of thing. So would we be able to implement that same thing in other countries? Like,
Absolutely. I think
so, yeah.
What it comes down to from for individuals in different countries is I think it takes, just like us, sorry, I'll need to turn that off. Just like us, you need to find some parents that are willing to put in the work. And that's really what it is. Like Nick just joined, he's the president of our foundation. It's Kelly, Kim, Katie, it's literally just parents that are putting in, you know, the extra hours after their day jobs are done, after their kids are asleep. You know, it's really, that's all that it is. So that's what I mean by community involvement. We need people. We're trying to work on, we have a full list of individuals that did respond to Kelly's Google collection. It's just even getting the time to comb through and to contact those people on top of everything else we're doing. We're struggling, guys. We're struggling. So we really need the community to step up and say, hey, I'm really good at this, or I have the time to do this. Like, let me do it. And if you're in Australia, it would be phenomenal if you created, you know, if you contacted your neurologist, your geneticist, and say, hey guys, can we work on creating a team of specialists here? You know, and that's all it is. Like, yeah, that's literally all that it is. It's parents just speaking and fighting.
I'm so pleased to have come on here today because I do work full-time as well. And we only found out not last year, the year before, that Grace has MED13L. So we've had, you know, 19, nearly 20 years of not knowing. So it's, you know, it's a bit sort of overwhelming going to the Facebook group sometimes, and you get a little bit overwhelmed by some of the things that are being talked about, and you sort of have been doing it a bit on your own. But I wanted to come on today and just, yeah, you've explained it so beautifully. I didn't really understand everything. I didn't really understand what the foundation is doing. Probably a bit like an emu with my head in the sand, trying to bluff that's not happening, because as you know, when you get the diagnosis, it takes about 12 months just to get yourself up off the floor. So yeah, if I know I can get in there and help and do some things, then yeah, I'm all for it. So thank you for explaining it so beautifully.
Thank you so much. Is James, I think he's there feeding his son. I think he's in Australia as well.
Oh, beautiful. Hi, James. I'm on the Sunshine Coast.
Yeah. Sorry,
Hi, James.
Also, just got up with the little man, so yeah. I've missed
No,
most of
that's
the
fine.
meeting, so I apologise.
Yeah, no, it's fine. I was just saying, you know, there is a community in Australia. So, you know, all it needs, you know, for people that are international is somebody who's going to raise their hand and kind of take the lead and be okay with, you know, knowing that at sometimes, you know, some people are in very difficult places with their children. And, you know, so it's not that they don't want to help. It's just, you know, as we all know, there's a lot of ups and downs with these kids. And so, you know, but again, just finding someone who, or for you, you know, to say, you know, that I'm just going to be the one. And, you know, what complicates things with us is because we do have an international community, but there's a lot of restrictions legally to collect specimens internationally, to transfer the specimens. So what we've actually done is we had one family come in from Spain or Portugal. I'm not sure where they came in from, but they came in, they were just coming in for a holiday. They went to Florida. So they connected with us. And when they came, you know, they sent the genetic report. We have a researcher who is heavily involved with us, who we had her look at it. And we said, is this child's specific variant, are they represented already in what we have? Because that's really important too. So don't think just because your kids' specimens or your kid's biopsy isn't in that study that Alicia's doing, don't think that your kid's not being represented, because a lot of our kids, even though they are unique, there's some similarities with the type of variants. So if your child has a deletion at like a midpoint or whatever, they're probably represented in Alicia's samples. So meaning just like that. So don't think that, you know, you're not going to be able to benefit from the information that Alicia's coming back with, and Dr. King's going to come back with, and, you know, whatever we're based, don't think that that's the case. However, something that I do want to say though, the more specimens we have collected, the more information we have. And that's probably like the most annoying out of all of this, is we're all pressed for time and everybody wants surveys filled out because they want to know what our kids look like when they were born, what happened during the development, you know, what were their challenges, what were their successes. They want to know, and they want to know regularly. And so as annoying that is, that's probably one of the most important things the community can do, because we need to have this foundation of, this is what our kids look like. And when we get to the point of where we have drugs to give our kids, we have to say, look, we gave them this drug and it's helping them. These kids usually at this age present like this, but now that this kid's taking this drug, they're not like challenged the way those other kids are. Do you know what I mean? So having that information on what is going on in our community with our kids, with their development, is so important. And we're getting ready to decide on, it's okay, Chris. We're getting ready to decide on a platform to go with for our natural history. We're doing our due diligence and like talking to every single rare disease foundation out there, what their experiences were, what they liked, what they didn't like, what the community liked. Did they find it annoying? Did they find it easy to use? And what we're looking for as a foundation is probably something I'm sure all of you would agree with, is we want the database to kind of work for us. We don't want to add more stress to any of our families. We want to be able for everything just to be in one place, super simple, and have the information there. I mean, there is going to be some work from all of us, but once the information's there and we regularly update it, again, if some biotech company decides they want to look at something for rare, because they want to make a big impact in a very, unique way, whatever, and they go and they look at all of the rare disease foundations, and they're like, oh my gosh, MED13L Foundation, they have all this information based on their mitochondria, all this information based on their fibroblasts, all this information based on their blood, all this information based on the history of these kids. They have mouse models, right? They have neurons ready to go. They have stem cells ready to go. They're going to be more apt to pick us to do their research on than a foundation that doesn't have anything. Do you know what I mean? So we're making it our mission to literally give them a toolbox and say, hey, come pay attention to us, do your science for us and help our kids. We're doing all of this stuff. So it's our job, and it's crappy because it's always the little people. It's always the parents that have to do the hard work. And then the people that come in with the money and the science, afterwards they get all the accolades, which I don't even care about. I'm like, you guys can have the accolades, just help my kid. Right. I'm sure everybody feels that way. So that's what we're doing. That's what this information with Alicia and the other researchers are really helping us do, is creating this toolkit where these, people are going to pay attention to us, because we're providing so much in-depth information. They can look at it and say, wow, if Alicia found this way where there is some mitochondrial dysfunction, maybe if we hit it with this drug, maybe it'll, improve the mitochondrial function, right? So we make it so it's simple for them. So we're doing all the hard work, but that's the goal. That's our drive, is to make it super easy for someone who can make a difference. Oh, Stephanie, what's the baby's name? Look at the baby.
This is Serafina.
Diagnosed the cutest baby.
Yeah, right.
Literally. Diagnosed.
I'm big girl.
You are a big girl. That's right. She's over here watching. So,
So because I just wanted to make sure that is there any other questions for Alicia regarding the genetics overview? And if you don't have them just yet, Alicia has a handout that we're going to be able to provide to the community. And then, of course, Alicia also provided her email. So if there's nothing right now, I just want to make sure that we thank Alicia and don't take too much of her time up. And thank you for Alicia for just the way that you explained everything. I wish way back when I received the diagnosis that I had someone to sit with me and kind of go through each part. So I very much appreciate that. We have a lot of newly diagnosed families, and I think that the way you simplified it and explained it was awesome. So thank you.
Of course, yeah.
So if you want to stay on and kind of chit chat about the ongoing research or how you can get involved and so forth, I just wanted to make sure Alicia knew that she could go to bed and get ready for her Monday morning if she wanted to. Thank you.
And maybe
Alicia, can
Kelly,
I ask
if
you
wanna
one more question? I'm sorry.
end the
Yeah,
recording.
yeah.
Just related to the mitochondria, I don't know if it's even related or if there's any research on it, but does the mitochondria and the production of what it does, is there any relation to that in seeing the hyperintensity flares within the brain? If that makes sense. I don't know if that's clear.
So we've only done in fibroblasts. We haven't done in neurons yet, but we have done mitochondrial output studies in fibroblasts and shown that it is decreased in MED13L samples, but we have not done neuron specific ones. But I know, as Vanessa mentioned and I mentioned, that it is all up and coming, so hopefully soon.
Yes, there is a group that the foundation just funded, the group at Vanderbilt, who Dr. Katie King is like the person to do this with that. So with the samples that our families contributed to Alicia and Randy's lab through Dr. Bain, right? And they've been able to transfer a portion of those cells to Dr. King at Vanderbilt, which is so cool, which is why participation and all this stuff is so important. So those 12 samples were sent over to, so a portion stayed with Alicia and a portion sent over to this other lab. And then a portion actually stayed with Dr. Bain, I believe. So they, now there's like 3 different places that are working on our kids. Do you know what I mean? So Dr. King's lab, she has the expertise to take the fibroblasts, turn them into stem cells, and then from those stem cells, grow them into neurons. And so from those neurons, they're going to do all the testing, which is specific to neurons. Because again, as Alicia mentioned at the very beginning, MED13L affects the entire body, so the entire system. So we need to look at what it does in the muscles, what it does in the cells. I mean, it's not even just muscles, mitochondria is in every cell, but what it does in the brain, right? And we're seeing, we actually have Dr. Gruder, who his specialty is what it does to the heart and the cardiac system. So we're literally hitting everything with everything we have. So it's just amazing people like Alicia that we're super excited to have in our life, that are really going to help us get to the bottom of things. Any other questions for Alicia? I do feel bad that I ramble. Sorry. I know I do. But sorry to keep you, Alicia.
No, I'm good. We're all in the same, all of us on the East Coast. It's not too late.
It's 7:17.
Yeah. It is not too late for a graduate student. Trust me. This is like, I wouldn't even be eating dinner most nights now. I'd be just getting home from the lab. So
thank you, Alicia, for
Thank you.
explaining it all. Yeah, amazing. Like Vanessa said, wish we had someone to sit down with us and point that out as soon as they were diagnosed. Yeah, amazing.
Mm-hmm.
And Alicia, we welcome you back anytime if you have updates or you think of something else that might help everybody understand a little better. You let us know. You're always welcome. You know that. You're part of the family now. And then we're going to continue having different Zoom sessions for family outreach. So if you have ideas, let us know. We have a couple scheduled coming up. And then we're going to have, Kelly had an idea just to have like some open free space where families can connect and meet. So we'll keep doing those, as long as everybody wants to attend and keep coming, we'll keep setting them up. And we really appreciate all the support the families are giving us.
Nick, do you want to say anything since you jumped on?
I've been listening to more than you know. Unfortunately, I had a conflict, so I had to multitask a little bit. But I'll just thank you to Kelly and Kim and Vanessa for organizing, Alicia for your time. And I have to say, Vanessa, you're amazing. You do such a fantastic job of explaining all this. So special thank you to you.
So true.
Well, thank you, everyone. And yeah, so again, as I mentioned, we're just parents too. Nick Seaver, not to belittle who you are, Nick, but he's also just a parent, fighting for his child. So again, none of us are disconnected, right, from the community. We are the community. So don't think that, we're up here and you are, you should even hesitate to send us a message, because we're in it too. So
we'll be sending more updates soon.
Yeah.
Yep.
All right, guys.
We're going to be posting the next Zoom sessions. We're trying to find times that match. I mean, obviously with people all over the globe, it's hard to find times, but if you if you get stuck and you feel like you can never get on, just reach out to us and we can try to shift a time, or we're going to try recording these and getting these posted for everybody as we go along. But just send us your ideas, things that you need to know more about, and we'll try to find an expert to teach us all about that topic. But thank you all for joining us.
Thank you. Thank you.
Good night.
Bye, everyone.
Thank
Good
you.
night.